We will spend the rest of the afternoon talking about quantitation, the tools that we have, some of the tools that has been there for quite some time, and we'll also, that'll continue on tomorrow morning with a new tool that's currently being built inside the TPP, and that's called St. Peter. And so that'll be talked about tomorrow morning. But so Luis will spend the rest of the afternoon with you, so he'll entertain you for all of that. So what we have now is the quantitation tools, and that's what sits in here. So as we've moved through these modules of doing the raw data into the database search engines, the profits, we'll talk about respect now, and so that is the ability to come back from the identifications and reanalyze those ones, and then you do this in an iterative cycle, and you'll see how that happens. But once you've got that, and if you're doing labeled quantitation, either through SILAC, is where you metabolically label your proteins from cell lines or other components. It could either be whole animals that have been fed with heavy labeled chow. They can be done that way. And there's even some experiments where I know humans have also been fed heavy labeled food, and they've gone and done some experiments that way. So there is a group, I know in San Diego in the US, that have had that approved for them to do that. Not that I would do that, but still. But if you're having other quantitation tools that you want to use, things like ICAT or iTrack, these programs will allow you to do that, and so Louise will go through those. And so really we have those tools here, and so XPRESS is really the calculation of abundances of proteins from isotopically labeled samples. And this is really the old ICAT component, so that's one of the ways you can get those done. ASAPRatio is also an alternative to XPRESS, but it allowed you to do a few more things. And so Louise will go through each one of those. Libra is if you're doing iTrack or TMTs, and so this is where you get the reporter ions. And so a reporter ion is when you couple a chemical component to the peptides, and if you have like 4 different mixtures, you can couple 4 different labels to it, and as it's getting fragmented, the reporter ion drops off, and that's what you quantitate again. So you're actually identifying the peptide through normal MSMS, but you quantitate on this reporter ion that gets fragmented off. And so Louise will talk about that as we go through. And the last thing is what we'll talk about tomorrow is this new non-labeling quantitation component called St. Peter, which is a re-implementation. So we now have those components all being developed and brought through at the same time. And so here is where we talk about these 2 options and how we can get these quantitation done based on isotopically labeled samples, and either chemically or through metabolic labeling. And really, it's trying to simultaneously identify and quantify all of these in your complex samples, and how do you actually get that with good accuracy and good dynamic range. And so Libra is also that other component. It's not the alternative to those 2 programs. That's a different type of quantitation. It's really based on those reporter ions going through. And then Louise, you'll talk about the issues with missing labels and things like that. Does that just drops things off? But it gives you both peptide quantitation and protein quantitation, so that's a good program. And then finally, St. Peter, which we talked about tomorrow, because we won't really spend much time on this today, is really implementing a normalized spectral count. And so it really takes in 3 different quantitations, so the peptide, the fragments, and also the abundance of those as well. And so it brings that together as a normalized function, so you can get that quantitation. That will be released as the new version comes out. So the TPP5 is a really major upgrade to a lot of the software that you've been learning. So as you're learning through now, you won't see much of the implementation in the programs you're using. They'll also still be there, but there's been a lot of work in the background to try and get a lot of the tools more up to date in terms of the software engineering, and that'll come through there. So with that, Luis, thank you.
Okay, so as Rob mentioned, we're going to start with this software component that we just didn't print it out on the schedule, but we've already mentioned it a couple times, called Respect. Again, here's my colleague David's picture. He's the primary author of this tool, so put his face there to remind me to give him credit for this. And the motivation for this is the following. So in MSMS, it's tandem mass spectrometry shotgun experiment workflow, where you feed in your peptides into your mass spectrometer, even though your precursor scan, even though you, the peak that the mass spectrometer chooses to fragment is very well defined, your selection window is actually much larger than that. So it is quite possible that even the highest peak is the one that's recorded in your raw file, in your MZML file. There could be other things that are coming into the fragmentation that are at different masses or very similar in mass. Another peptide, for example, that it happens to fall within that selection window. So in this case here, this is just one type of peptide. And these are the different isotopic components of that peptide. And so this gets fragmented, and you get your MSMS scan from the fragmentation of this one peptide species. But in some cases, it could be that you have some other peptide that is very similar in mass. So in the precursor scan, in the M over Z here, is very close in mass to that one. And some of its, either all or some of its isotopic components might actually fall within that selection window. And in this case, the selection window will include two peptides, this blue peptide and this pink peptide. And so when you fragment that, you'll get, at least for this example again, you'll get very strong peaks from this very high intensity signal peptide, but you may also get fragmentation from the extra peptide species you're getting into your selection window as well. And we call these chimeric spectra. A chimera, it's an animal that is composed of multiple animals at one time. So this is the name we give this. And how do you actually get chimeric spectra? They're fairly common in tandem mass spectrometry, and it's somewhat proportional to your sample complexity. So if you have a lot of peptides in your column, and they're also overlapping in time as they elute, you're very likely to get that. It's also proportional to the selection window size that you choose for your mass spectrometer. A very narrow selection window might isolate your precursor better. A wide selection window will tend to then include it'll be more likely to include other species in it. And obviously this is influenced by your chromatography. So, your buffer, how your fractionation, etc. And by default, the vast majority of database search algorithms are designed to only report one peptide for each spectrum. They're not really designed to find two or three. There's probably a couple search algorithms out there that try to do this, but pretty much all the standard ones in general only look for one peptide. And these are some of the reasons why. So the search algorithms do not find chimeras because the selection window of the instrument is much wider than the peak resolution. The algorithms, when you use Comet or Sequest, Mascot, they use only the measured precursor mass within your tolerance that you pick in your search parameters, such as 10, 25 ppm, especially in high mass resolution instruments. You tend to specify a fairly narrow mass tolerance that is much smaller than the selection window of your mass spectrometer. And in this case, then selection window may include peptides that are outside of the search mass that you use. For example, if you have a peptide of a different charge state, like a charge state 3, and then another one charge state 2, that one may fall into the mass range, M over Z range of this mass window that you never considered because you were not even looking for that one, given the precursor mass that you were looking for. So that's one reason why Comet will have not found that other peptide. It was never in its search space. And so here's just an example of how that works. This is similar to that other cartoon I just showed, where you have this, the selected ion, as reported by the raw file. The mass is 615 m over z, and of charge state 3. That was determined by the mass spectrometer, and there's the isotopic envelope of it. This being charge 3, if you do the math and add whatever the proton or something, it means that the database search was done on peptides that were of mass, of total mass, 1844.22 Daltons plus minus your mass tolerance. So when Comet ran, based on the precursor mass and charge, it was only looking through the possible peptide sequences in the database that matched this mass plus minus a small tolerance. But you could have a chimeric ion of very similar m over z. Remember, m over z is mass to charge ratio. So instead of 615.74, 614.85, still within the mass selection window of the instrument, but it's only at charge 2 in this case, which means that its actual neutral mass is 1227, and there is no way you would have even tried to search for that when you were really only looking for peptides of about 1844 mass. So it would never even be looked for, searched for. So this tool that we've developed, and mostly developed, called ReSpect. So it's like re-analyzing the spectrum, and the word respect, so it's kind of funny. This tool allows you to identify chimeric peptides from given spectra. And because it's part of the TPP, it uses the PepXML and all MZML formats. It works with all of our existing search algorithms that TPP supports. So it could have been a Comet search, could have been a Spectra search, could have been any other search. It's all compatible with anything TPP is compatible with. So the approach is the following. So you do a normal we call it first pass search, your standard search, where you use whichever search algorithm you want, or multiple ones if you're doing iProfit, but say Comet using the standard narrow mass tolerance and charge states that come from your MZML file. You take those results and run them in the typical TPP fashion through the statistical validation tools, PeptideProfit and iProfit. And what then ReSpect will do is we'll go through each of those identifications, and you can tell it a threshold of probability to actually look for. And it takes, and I'll show this graphically, it will take the spectrum, the original spectrum, and remove, and actually not remove, it will attenuate, cannot say this word, it will reduce the peaks in the spectrum that are matched by the first pass search and proportional to the probability given. So probability of one peptide will have all of its peaks removed from that input spectrum. Probability 0.5, those peaks that match will only be reduced by 50%, etc. Once you remove those or decrease those peaks, you save that new spectrum file into a new MZML file. And then you repeat the search on that new MZML file that has now modified spectra using maybe a wider mass tolerance and assuming all charge states. So at this point, you don't assume what charge state and what the precursor is, and you expand your mass tolerance so that you're able to include basically the size of the selection window. And the results of that are, of course, then validated, so make sure you actually got good results through the standard tools. And then you can do this over and over again. Each time you match a peptide, you reduce the peaks, you save that new spectrum, you search it again, you validate it until you are left with no spectrum to analyze. I'll show you here graphically how this works. So with respect, this is the original spectrum, some given spectrum where you match the B and Y ions quite well. And there they are in the spectrum viewer. And the original identification, the first pass identification of this, after the search of this and validation, was 0.999. So you take all these peaks that were matched and reduce them to 0.1%, right? That's 1 minus 0.999 is 0.1%. You get this. And you see that now, that same sequence doesn't match any peaks. There's still some peaks here. You can barely see them. And maybe in your slides you can see that there's some peaks left. But now you've removed the ones that matched the first time. You take this spectrum and you save it. And then you search it again. And so the peaks that sorry, let me go back. Going back to this, these peaks look small now, but it's because they were dominant peaks. You know, these peaks were so tall compared to the little peaks that are left. So once you remove these big peaks, you can then see that, you know, these peaks were there all the time. They were just very small. So we remove those ones that were matched the first time. We do a second search, and we find that this new peptide matches with very high probability many of the remaining peaks. You can see it's a good match just based on the ion series there. A lot of B and Y ions that are matched. And indeed, peptide profit, after the search and validation, still got 0.995. That's a very good probability match. And then you would keep doing this. You would remove these peaks and then do it again until really you don't get a good result. So here we show you the mass differences of the results after running respect a couple of times. And you see that on the first pass search, you know, the first time you do the search, you typically use a very narrow mass window. We talked about this on the search day, and when we do the tutorials the next couple of days, we'll see how to set up the parameters to do, you know, a standard narrow mass window. You'll see that the search tools allow you to not only specify a narrow mass window, but also they automatically can look for the same narrow mass window at isotopic offsets. And you'll see why this is very actually powerful. But you'll see that after a first pass search, all the peptides you find are in these mass ranges. After a respect analysis where we open them, you know, we reduce the peaks and then open the mass window and allow for other charge states, you see that now we actually find peptides. The majority of them have the mass difference between the precursor and the identified peptide. Yeah, most of them are 0 difference, and many of them are indeed at isotopes. But you also find, you know, just peptides are in between mass, and those are by and large different charge states. That, you know, they come from a different charge state. And so, and I already touched on this. So the chimeric depth. So a chimeric spectrum may contain more than 2 precursors. So we're not assuming that there's just 2. There could be 3, 4. Who knows? So as I showed before, respect can be iteratively applied until any new PSMs are no longer found. And this shows you yet another example of this. So this is the original match. After the first pass search, you match all these peaks. You see that it has very good coverage and very good ion series. There's a few peaks that are not matched, but overall this is a very good identification. After you remove those peaks and redo the search, you'll see that, these 3 peaks here, they were right here. It's just hard to see, and they're grayed out. So over here you see that the second pass, we identify this peptide, very good match to the ion series. Remove those peaks. Second iteration, so the third peptide found in the same spectrum, found all those peaks, then you remove them, and even a fourth peptide, so the third iteration, and you still find a fairly good peptide. So one spectrum now, in this specific case, we found 4 peptides with high probability, you know, and good match. And so this is part of the paper publication. This is unfractionated yeast sample that we had ISB, and you see this represents the number of PSMs that were matched in the first pass. This is the second pass, the third, and you see by the time you get to the fourth pass, you really only identified only a handful of things. So it's not like it keeps going that much. This was almost 400,000 spectra, and by using respect in this manner, we got a 30% boost. We found 30% more peptides. That's pretty good. The same analysis, but for a different sample, where you have just medium fractionation, where instead of being this, this 4 replicate yeast, unfractionated yeast runs, this is from published IPRG 2013 study, only have 100,000 spectra. And these were actually, so it's fewer spectra, but it's also 14 fractions. So this is a more highly, more fractionated sample. And you see that in this case, the original pass, the original search had a lot of IDs. The first respect search had only a small portion of that boost, and you only boost here by 8%. So the amount of extra IDs that you get in this case is only 8%. And in this, again, in another study within the paper, we see that in this case, this is a highly fractionated 48 fraction study with in Orbitrap Velos. You see the first pass results are now on this side. First respect, by the time you do the third respect, you don't find that many more. It's a 13% boost in this case. So that's still pretty nice. So this just shows you that for different data sets, you have different amount of chimeras or chimeric spectra. And the biggest factors in this are the sample complexity, as we talked about, and the instrument settings. And here we show you, we did a few of these, and we have plotted here the ratio of distinct peptides identified on the first pass versus the percent boost after using respect. And you see that for these unfractionated samples where we have very few peptides identified the first time, we get a huge boost by using respect. People may ask, and rightly, so do you get more proteins? So you just because you get more spectra identified, does it help on the protein level? And you see that here's an example with this the yeast, the unfractionated yeast sample, and this is protein profit predicted and based on decoys, and you see that at the same error rate, see that there's 1% error rate, we're getting about 600 proteins, and after respect analysis, now we're getting about 650. So again, about 10% boost in proteins. It was about 30% boost in PSMs, translate to about 10% in protein. But this just shows you how to run it and the couple parameters that it has. It has after you select your input files, your PepXML files, you can specify a minimum probability to cut off, and also the mass tolerance to match the ions in the MS2 spectrum. So just as a summary, what respect does, it will actually take the peaks that were already explained by the first pass match and reduce them based on the probability of that match. That produces a new spectrum, and actually a new spectra when you take many of them in one run, and that creates a new MCML file that you can now research. So search again with different mass tolerance, maybe a larger mass window, and assuming all charges possible. And obviously we use the same validation steps of the peptide profit and iProfit to estimate the error rates of these new identifications. And because it is part of the, it uses the same formats, and it's all integrated in TPP, so this is all not really automated as in one button does respect multiple times, but you can then just select the MCML file, select your parameters, and the output goes into PepXML, which can then be run through the profits. But it also works just fine on standard mass accuracy MS2 data. We tried with LTQ data, and it worked just fine. It just works better with better data, like everything else. So let's move forward, and as Rob already introduced, we're going to now talk about, spend the rest of the afternoon talking about quantitation. So here's our roadmap here, and you see where the quantitation is done. Quantitation is done at the peptide level, which obviously then will lead to values at the protein level as well. But we quantitate peptides, quantify peptides. And this first lecture will be based on isotopically labeled samples. And for isotopically labeled samples, we have these two programs that I'll describe in the next few slides, XPRESS and ACP ratio. The other program that I show here, Libra, is for a different type of labeling technique, and we'll talk about that in the next lecture. So for isotopically labeled samples, as opposed to isobarically labeled samples, we use these two software. So here's the lecture outline. We're going to talk about the principles of quantitative proteomics, and then we're going to talk about peptide and protein quantitation with XPRESS. We probably already covered this a few times. This is just a summary of shotgun proteomics. Protein mixtures digest into peptides. These peptides are concentrated and fractionated by any number of separation technologies, such as isoelectric focusing, reverse phase chromatography, etc. As they elute from this column, peptides are ionized and analyzed by tandem mass spectrometry. The identification of those peptides, the identification comes from the CID, from the fragment spectrum. The MS2 spectrum is what leads you to identification. But for this technique, for quantification, that comes from the MS1, from the MS signature. Again, except in the case of iTrack. So here's a cartoon of that. And there are some complications with this. So as we now know, shotgun mass spectrometry detects peptides, not proteins. We take PSMs, and then we assemble the proteins from that. And because of that, we also have this issue, as has been brought up, that there could be a peptide could map to multiple proteins. So how do you resolve that? So that's something to keep in mind. Strong cation exchange chromatography has good but not great separation power. And also the same peptide species could be found in different fractions. So if you're doing a fractionation, that same peptide could be in several places, and you have to deal with quantifying that. When you do reverse phase chromatography, as you elute out of your column, you have different peptide species eluting at any given time, and you'll get a peptide elution profile. Say as a function of time, as you're pushing all these things through the column, you'll get some type of curve where the peptide's coming out, and then when it stops being there and go back to zero. But this one peak, it's a little misleading because that's what's coming out of the column. But when you ionize that eluting peptide, this could actually take multiple charge states. In this case, we're saying 1 to 4. It could be more. Sometimes we get enough charge state 5 out there. And so you see that even though there is only one elution peak, that could create in your MS1 map at different charge states, different amounts of different chromatographic peaks here. And obviously the size of this will depend on the what ionization state is more preferred by or more likely to happen for this one peptide. Ideally, you want to take advantage of all those, right? So you want to keep that in mind as well. For the thing you can do is that, again, this is the MS1 as it's eluting. Could be that this peak here, this very large peak, got fragmented. The instrument decided to do CID on this, that's the star, and confidently identify the peptide, and it knows that the mass and the charge state. And because it knows the mass and the charge state, you can actually calculate what the mass of a different ion would be. So if this is +3 and it was identified, you could go back and say, hey, well, I didn't identify the +2 charge state, but where is it? And you can find it, and you can see maybe there's a peak there. Likewise, for this other peak, it was probably it was very, very, very strong peak, and it was identified as a charge 2 peptide of a certain mass, you can then calculate what the mass would be, the m over c would be of its corresponding triply charged ion. And perhaps you find a signal there. And so in order to quantify peptides, it would help us if we actually can find all of these ionized states. So again, just to remind you, peptide identification, you match the CID, the MSMS spectrum with the database. You could have multiple IDs for the same peptide. So different isotopes, light and heavy. So you have a Silac label, for example, you could identify the peptide in either or both of those states. You could have different charge states, 1, 2, 3, 4, etc. Or you can have repeating IDs. So the same peptide ion with the same modifications could give you multiple spectra and for the same charge state. And so you want to try to use all this information to quantify. The way we do it is then, I think Mike showed this as well. This is you reconstruct the single ion chromatogram. So you go back to the MS1, and this is just the 2D view. At any given time in MS1, you have intensity and m over z. And then if you look through time or scan numbers, you get this curve, where you see at even given m over c, you can pick a time and just cut across. Sorry, given m over c, you can pick a m over c and then just cut across different time and look at the area under the curve of that. So this becomes this curve right here. The area under this curve, this single ion chromatogram, is proportional to peptide abundance, but there's a problem. There's actually a few problems. It could be that one peptide ionizes more efficiently than some other peptide. So that area under the curve is not just only a function of the abundance of that species, but also how well it ionizes. And we actually don't know how this one peptide responds to certain ionization conditions to this other one. One solution to this issue is to then have two samples that are chemically labeled with different isotopes. So you have a reference sample and then a labeled sample that you can compare against. A stable isotope renders the new peptides chemically identical. Peptides, you identify them before you quantify them, so you still get the MSMS spectrum, and you can run your standard search and other validation algorithms to find the peptides. But you can distinguish these two samples based on the in MS by their mass shift. So you're adding an isotope that has a different mass, but it's just more neutrons, so it has the same chemical properties. And the ratio, the peptide abundance ratio, is measured by the ratio of the two areas in the light and the heavy. So we call these light and heavy when you have two isotopes. As Rob mentioned, there are different labeling methods. So you can have metabolic labeling, where you take either your plant or your mouse or your favorite human and feed them, put them in an environment where they acquire a special isotope of either carbon or nitrogen, and you eventually hopefully get full uptake in that, at least in the place where you want to study. Or do an enzyme reaction, like at the time that you apply your enzyme and incorporate oxygen 18. In either of these cases, wherever if you incorporate at the metabolic level, at the chemical reaction level, or at the enzyme level, you are going to get peptides from two conditions that some have the light version of the tag and some have the heavy version of the tag, but they're otherwise identical peptides. When you run through the mass spectrometer, you will see the MO in MS1, the peaks of those, you'll have pairs of these shifted by that mass that you incorporated. And so just as a summary of this, so your in this case, two samples. There's now techniques by which you can do more samples of this type, but they're still kind of nascent. The samples are isotopically labeled, so chemically identical, just different mass. And you can simultaneously identify and quantify thousands of proteins in complex samples. You must at least identify at least once a peptide ion in the MS2 spectrum so that you can quantify it. Otherwise, you just don't know what that is. It's stated here that the accuracy of this method is it's not great, but it's decent. It's 10 to 30% if you so do it all right, with a modest dynamic range of about 2 orders of magnitude. Now, because we go from spectra to peptides to proteins, there's a bit of a hierarchy structure to determining a abundance ratio. And so if we start here at the, we're going to work our way back up. The CID is the most basic level. This is the actual spectrum that we get. This is the MS spectrum where we identify peptides. And so at the CID level, we could maybe quantify from this fraction, the heavy +2, or maybe from this other fraction, the light and heavy in +2 and also the +3 charge state, perhaps a different fraction over there. And these two correspond to the same peptide, so now we'll be quantifying this in this fraction, these two in this fraction, and then getting a unique peptide ratio. On this end, we would get a peptide ratio for this one, and they will be combined to get the actual protein ratio. So you're going from peptide IDs and ratios all the way to, of course, identifying the protein and getting its abundance ratio. And the two softwares use slightly different statistics to do this. So XPRESS, there's a publication. It was the first, so my understanding, I wasn't around at ISB in 2001, but the Ebersol group created, I think, the first isotopic label, the ICAT, the cleavable ICAT label. And because they sort of developed this technology, they also needed the software to do this. So Jimmy Eng, the same person that wrote Sequest, mostly wrote this software, XPRESS, and he still maintains it. So XPRESS is actually still maintained by Jimmy and still worked on a little bit, even though it's also one of the more legacy programs that we have. XPRESS, to calculate its peptide ratio, it goes into the it reconstructs a single ion chromatogram of the charge state in which the peptide was identified. So if the CID was done in charge 2, it will only do the charge 2 for that one. It will not try to go and match the charge 3 and 4 and 5 and 1. Once it pulls out all those data points from the MZML file from the MS1, it does all these programs. It will do a basic smoothing of that. It becomes very jagged. All these peaks are pretty jagged like that. So you need to then smooth this out a little bit to be able to then fit a curve around them. Done using this type of filter. But the protein ratio, XPRESS calculates the protein ratio as the geometric mean of the peptides. So in a normal average, in a normal mean, you just add things and divide by n. In a geometric mean, you give more weight to things that have more signal. So you take the logarithm of each ratio, sum that up, and make an average of the logarithm, and then raise 10 to the power of that. And that is the ratio at the protein level. That's just the way they decide to do it. And there's also an estimate of the error, basically doing standard deviation on that logarithm. And once this is all done, there's a page that you can access via the appropriate link, where you can see for any given protein, all of its peptides and all the ratios of those peptides, and with links to go and actually interact with the results. Once you do have the results and you've run the software, again, this is the PepXML viewer that you have yet to meet, but this green thing in the background is the PepXML viewer, and there will be, at the peptide level, a column that shows you for that PSM, for that peptide, its ratio, and it's a link to that page that you saw in the previous slide that looks a lot like that, that will show you the quantified peaks and ranges in scan, the scan ranges that were quantified. And an interface to curate that, and again, that's the tutorial in a couple of days. That's at the peptide level. At the protein level, when you have the protein output, there's also a link that takes you to a page that shows you all the peptides for that protein and their ratios. AC-peptide ratio is somewhat similar, but it actually goes a lot further. It tries to do a lot more. Again, this is our hierarchy that I showed you a little while ago, where at the bottom we have at the CID level, a ratio at the CID level. So if we identify a peptide, we go back and look at reconstruct the chromatogram for that ID. We go back to the MS1, and we get a CID ratio. Then we go back, and from these ratios, some of these will be the same peptide, just in different charge states for the same fraction. So we then have a LC peak ratio, so per fraction. And then in some cases you could have maybe two different fractions, and when you combine those you get the unique, we call this unique peptide ratio. So if this peptide ends up in multiple runs, you combine those ratios from the different runs and get just one single ratio at the unique peptide level. When you have n number of these peptides that map to a single protein, you will combine all of their unique peptide ratios to get one protein ratio. So there's a hierarchy of analysis that the software is doing. So ACP ratio works in slightly similar way as XPRESS. It will, of course, go back and reconstruct the SIC for each of the identified peptides. Step one here, how does it reconstruct the SIC? Well, you always assume that the search result is correct. Otherwise, well, it's a non-starter. So you assume Comet did the right job. This is the right match to this to this spectrum. And you go back to the raw chromatogram and opens an M over Z window, so like a mass tolerant window of a certain size, and you reconstruct over time the chromatogram. This is a parameter that when you run, you can specify the size of that window. A very large window will start including maybe other isotopes or other species. A very narrow window may not have enough signal. So you sort of play with that depending on your experiment. The smoothing, because apparently a low-pass Butterworth filter was not good enough, this one uses a Savitsky-Golay smoothing filter. I think I was going to write a web application where it just throws two Russian names together, so you can name your new algorithm that, because every time you put two Russian names together, it sounds like it's a really important thing. So Savitsky-Golay smoothing filter, just to again to smooth out the signal. And in this case, ACP ratio will try to calculate a background over that chromatogram and subtract it. This increases your dynamic range. If you take away background, then you have more signal that you can actually identify and less error. This is just what it looks like when you go to the interface. And you can see why we need to smooth things out. You see the raw data is this, the red line is the raw data. And when you smooth it out, you get this blue line. So that's a bit more of a peak as opposed to all this jagged stuff. The pink line here, right here, that's the background, and you see it's been subtracted. And the amount of signal being used to quantify is this green line, and you see it doesn't take into account the background, just the stuff right above it. And actually, in this specific case, you can tell that the software, in its smoothing, sort of missed half of the peak here. The smoothing found a valley here, and so it stopped integrating there. And this is one reason that you have to go back and, for proteins that are interesting to you, you want to go back and make sure that the ratios are well calculated. All software is bound to fail in some way or another, especially when determining peaks is not always that easy when you have overlapping peaks and whatnot. So it tries to do a good job, but you can see in this example I'm showing you, they should have quantified all the way to the end of that. And as I mentioned before, peptides will be ionized in different charge states. You can see here out of 1900 peptides, you see that for this specific case, it's an older slide. There are many that are in charge 2, but they also are found in charge 3, 4, and 1. And this is how many of these peptides are found simultaneously in all of these 4 charge states. See 1, 2, and 3, not that many in 4. And so ACP ratio will do the quantification, not just for the charge state where the CID was made, which in this case is charge 2. But you see here we have tabs that show you charge states 1, 2, 3, and 4. The little bracket means that there was no signal. But 3 and 4, when we go there, you see that there was signal there. There was no CID made there, but we went over, the program went over there and found a signal that was able to quantify in charges 3 and 4. So he incorporated that into, and so each of these charge states have a ratio. This is 1.2, 1.2, 1.3. Combine them and you get the unique ratio. That is actually not shown here. It's above in this page. Whenever you don't like a specific charge state or ratio, you can just turn the acceptance to no, and it will just not include that in the calculation. Again, we'll be actually working with that in a couple of days. But you have a lot of control as to what happens here. So in order to calculate the peptide, unique peptide ratio, you evaluate the ratio with an error from each of available charge state. You remove any outliers. Sometimes, one of the charge states might just be way off from the other ones. It was just a identified signal. And so you use this Dixon's test, simple statistical test to just throw out things that are not, that are clearly outliers. Unlike XPRESS, you will do a weighted average. Instead of just a geometric average, you do a weighted average. So each of the charge states is weighted by the chromatographic areas. So the more signal you have, the more confident you are of that, ratio. And so you do a weighted average and you use the area under the chromatogram to do that. And you do standard statistical methods to calculate the ratio and the error standard deviation. And so here's what would show above this. You get for this specific spectrum and this peptide, I guess usually it shows you the peptide somewhere here. You see that the ratio of that light to heavy is 1.2 plus minus 0.07, which represents 6%. So this ratio has an estimated 6% error. So that's pretty good. That's actually quite good. So far we've done this first step here of the peptide ratio. Oops. So step 1 is to do exactly what we've done here. So that was the first peptide ratio. Now we want to calculate the unique peptide ratio. So there's 2 steps. Step 1 is to group these ratios of the same peptide and the same peak together. Different isotopic forms, different charge states and repeats. Many of them will be the same. If it's the same peak, you're going to get, it's the same elution, so you get the same ratio. But if they're not the same, as I just mentioned, we do a weighted average based on the chromatogram areas, and we calculate, we throw away outliers and calculate the mean and standard deviation. Step 2 is to group the abundance ratios of the same peptide from different fractions. Again, this is if you have different runs where you identify the same peptide across different runs, you would, you have an MS1 spectrum for each of those runs. So you would quantify each of those and then combine them here, again, using the same technique. You would weigh the data points by chromatogram areas, and you calculate mean and standard deviation, and you throw away outliers. And so here's an example. We had to compress too much information in this slide, but you see that for this one peptide, it was found in 2 different fractions. And so each of these fractions, see this one, it was found once, and it was quantified there. In this other fraction, it was found 3 times, 3 different times. And so it was quantified in all those 3 different times. And so the ratio, the average of these is there. Well, that one is only once, and then the average of these 2 things is that ratio. So this is the CID ratio, the peptide ratio, and then the unique peptide ratio, which is that one. All these things are links, so you can go and actually look at the chromograms of each of those and evaluate whether they are correct or not correct. Again, because of if this was an outlier, if any of these was very different from the others, it would automatically be rejected by the software. It would have a no, and it would not become part of the average. but you are able to override that by actually going in and changing that. And the next step, well, almost the final step, but the next step in calculating is to calculate a protein ratio. So now you have the unique peptide ratio. Well, now it's actually fairly simple at this point. You get all the peptide ratios of the same protein, throw away the outliers. You do also a weighted average based on their error. So this, again, standard statistics. A large error carries low weight, a low error carries larger weight. And you calculate the mean standard deviation of the ratio for that protein. And so here's an example. Here's what the screenshot would look like at the ACP ratio page for a protein. You'll see that this protein has, in this case, 3 peptides. 2 of these peptides have kind of similar ratios, 2.3, 1.9, almost 2. This other one has 0.54, so the software determined this was an outlier and automatically rejected it. And so the actual accepted protein ratio is just the average of these 2 based on their error. They have similar error, 12% and 10%, so it's almost a straight-up average of that. And the actual final has 14% error. So just to summarize ACP ratio, its features. Just like XPRESS, it can handle any isotopic labeling technique. Again, no iTrack, that's no isotopic labeling. It can estimate the errors at the peptide and the protein level and the ratios. It can calculate ratios from multiple charge states, not just from the one where the CID was matched. It can take into account the signal background, and this increases the dynamic range. But you're able to zero that out or change that value as you curate this. I'll just show you really quick how ACP ratio is run and the output. And again, a lot of these slides will serve as reference when you're either doing the tutorials or running the software on your own. You can always, in Analyze Peptides tab of the Petunia interface is where you find peptide profile and also ACP ratio, all the peptide level analysis tools. If you scroll down that page, you will see that there's an option to run XPRESS and also an option to run ACP ratio. You don't have to run both at the same time. We will, just because we will be doing a tutorial. Some people choose one or the other. Some have preference, a preferred one. Again, Jimmy Yang only uses XPRESS. I don't think he ever touches ACP ratio. But other people prefer to use this one. Some people use both. When you have a protein profit output file, you can find, for each of these protein entries, you will find a section where you have the ACP ratio value and also the XPRESS value. And the ACP ratio will be reported as 3 numbers. One of them is the raw ratio. Another one is the normalized ratio with an error. So after you normalize based on the sample dependent normalizing, and a p-value based on the profiling of that. So 0.93 is not really an interesting p-value. And you see that after you normalize, the ratio is about 1. So there's, that one specifically is not that interesting. This one down here maybe is a little more interesting because it was 1.06, but after you normalize only 0.77, and based on the profiling, the p-value is 0.6. Well, still not great, but still more interesting than the other one. Oh, and so that's the blow up of I always forget that one comes right after that one. So now you can actually read it. So there's a ratio and the standard deviation. Tells you how many unique peptides it used in the calculation for that. And the normalized ratio with the standard deviation and the p-value. If you click on that link, you get to the peptide ratio for that, again, for that CID. And you have the different charge states that you can look at. So there's charge 2, charge 3, charge 4, and the different, we'll be playing with the different ways of interacting with. You can make changes in this interface. You can change the background level. You can change the scan range because maybe you got it wrong. Maybe you like a different scan range. You can change the background. Once you make these changes here, you can click on this button that says, OK, evaluate the ratio. Let me know what the new ratio would be. And when you do that, you get a new ratio that if you do then like the new ratio, you can then save it. And you can do that at the peptide level, and also once you do each of the different peptides, you can curate at the protein level in the same way, where you get a ratio, a working ratio as you change the different parameters, and when you're happy with that one, you can save it and save the ratio at the protein level. So how to interpret results? Probably the best, the easiest thing to do. If you have thousands of proteins, you're not going to go through each one of them, unless you have a lot of time or a lot of money for postdocs to do this for you. So the recommendation is that you sort by this p-value. P-value will give you, if you sort by that number, you will get those ratios that are the most significant, either up or down. And once so that gives you the potentially interesting data, but that's not enough. Maybe the ratio is wrong. Maybe it has a ratio of 200 just because there's some trouble. So you want to go in and identify and verify troublesome peptide ratios. So in this case, you see that of these 4 peptides, 3 of them have a fairly similar ratio, 1.2, 1.7, 1.3. This one's 4.4. Well, that's very different. And it could have been thrown away by the Dixon's test or not, but you still want to maybe look at it and see if there's something there. And this is just showing you then how to interact with the software. Again, we'll be doing this thing manually in a couple of days. But if there are peptides in the same LC peak in the same experiment, you can just analyze one ratio. The other ones will be very similar, so you can just reject them or not worry about them. You definitely want to pay attention to unusual data, such as something that has a very large error. So something that has 100% error is probably something you want to look at. Also, in the case of infinity, like 0 to 1 or 1 to 0, or unknown, sometimes they just can't determine a peak. You may want to go in and see that the software sort of did the right thing, or if you can still save a ratio out of that. Done pretty quick here. After we're done, we'll actually then go back and do things with the TPP installation. So we'll actually use all the time we have. Okay, so I just spent the last almost hour talking about, isotopic labeling quantification. There's another way of quantifying, labeling your sample, and this we call isobaric labeling. And this is techniques that you can use with these reagents like iTrack and TMT. How many people use iTrack already? A couple, yeah, okay, a few. Anyone using TMT? Oh, you use both. Wow. Great. Again, in the roadmap, we're still in the same spot here in quantification of peptides and then obviously proteins. And the software here is Libra, is separate from ASAPratio and XPRESS. It's not an alternative. It's actually a different technique. So this is the overview. Talk about MS2 level quantification. A little bit of background on iTrack, and then how we quantify using the software at the peptide and protein level, and how to run and look at results. So in the case of iTrack here, chemical reagent that you buy a kit, and the original one was actually 4 different reagents. Now they, who is this now? SciEx sells up to 8. But it's still otherwise a very similar technique. These reagents all have the exact same mass. All 4 reagents have the same total mass, and you attach them to your peptides. I actually don't know how, because Rob doesn't let me touch anything in the lab, or the mass spectrometers for that matter. But so you attach this entire thing to your peptides that you want to quantify. But you have up to 4, or actually up to 8 different kinds. You are now able to quantify. In this, I'm just stick to iTrack 4 different kinds of conditions. So when you get a spectrum, an MSMS, so now this is tandem MS, fragmentation spectrum from a given peptide that has these tags. The spectrum, the B and Y ions will come from, say, up to the 4 different conditions. But if you look here in the low mass range here around 115 or so, and you blow that up, you see that there will be 4 peaks, ideally, each corresponding to the abundance from each of those conditions at the 114, 115, 16, and 17 mass ranges. Again, these peaks come from this reporter portion falling off, getting with a charge and getting. So Libra is the software that will do this for you. It will parse the MZML files to extract the intensities, a list of values that you tell it to look for. The case of Libra, we have to actually, the parameters for Libra are stored in a file called a condition file. And so we need to generate that before we run Libra. And you can generate, at the moment, this file for up to 8 channels. There's one, so TMT is sold in 6 or 10 kits, but TMT10 is actually using some very narrow mass differences for 3 of the ions in the middle. So this tool that we have in TPP to generate the condition file is not able to generate those tiny difference masses. So these numbers you enter, and you'll need to see this. This is one of those XML files where you enter this. Again, Petunia can just generate this for you if you fill this out. You don't have to fill it out necessarily. Petunia already has the defaults, the last known that I know about. So if you select that, it'll just automatically fill it out for you, but it's up to you to make sure those are the right values. Another parameter that you may want to adjust is the tolerance. The M/Z tolerance is just how wide a window are you going to look for your peak. And to make it simple in Libra, we just take, within that window, we just take the highest peak and quantify that. We don't try to average out any other peaks there, because they could just be spurious fragmentation peaks that don't have nothing to do with the eye track peak. So whichever one is the largest one, we use that to quantify within the tolerance window. There's an asterisk here that if you're using TMT10, you have to use an extremely narrow mass tolerance, because the difference between in TMT10, there's a few reagents that are really close in mass. We have run actually Jimmy has run the analysis on TMT10 using Libra, and it works fine. We just can't generate the file with the intensity corrections yet. But they ran it without intensity corrections, and it seemed to work fine for them. So it can be done. And the last thing you may want to do, the last important thing you may want to do is centroiding. So if you acquire your data in profile mode, your peaks will look something like this. And so you can ask Libra if you can centroid at any step along the way. Typically, people do centroiding when you convert data from raw to MZML. You can do the centroiding there. Basically, it's called peak picking. But if you didn't do that at the time, you can tell Libra to just do either mathematically, like an average or an intensity-weighted, center of mass, or just no centroiding, in which case it will just pick the highest point here to quantify. Otherwise, if you centroid, you take all that signal and put it in one. Peaks that are incoiluded? You mean like, so are you talking about, again, like chimeric spectra, or? Kind of. Yeah, we haven't, so yeah, if you have 2 peptide ions, and they're both I-track labeled, all fragmenting at the same time in one, yeah, I guess obviously we can do respect to identify the other one, but I don't know, yeah, we don't have any way of rejecting the ratio or anything for that. A couple other parameters you can ask, you can tell Libra which channel you're normalizing against. If you have 8 conditions, which one is your baseline, you tell it. By default, the target MS level is 2, but you can do this whole other MS3 thing. If you're doing fragmentation, the MS3, you can tell it to look for the fragment ions there, and some output options. Lastly, almost lastly, by default, Libra will only quantify peptides that are above 0.5 probability. It's basically not bothering with low probability peptides, since they may be wrong. Anything below 0.5 means it's more likely to be wrong than right. So That's the definition. Another caveat of this is that in order for it to quantify, all channels that you're looking for must be present to quantitate. So if you're doing a iTrack 8 and it only finds 7 peaks of the 8, it will just declare, I can't do a ratio. At the protein level, it's doing a very basic outlier removal. Anything that is more than 2 sigma from the mean in the ratios gets thrown out. All the peptides get thrown out. All peptides beyond 2 sigma from the mean are thrown out, and then you recalculate the mean and the standard deviation. In the standard deviation, if you use only one data point, one peptide, by definition, you have no standard deviation, so it's reported of 99.99. So if you see that number, that means, oh, you only use one peptide. If you get -9, it's just the code it has to say that after outlier removal, there were no peptides left. So there was some signal to begin with, but when we removed outliers, I was left with nothing to quantify and get a protein ratio. So just things, again, for reference once you view the results. There's a little bit more information. There was never a formal publication, but there's information at this page that describes this and a little bit more of the software when it was first written. In order to run Libra then, again, this all done at the peptide level. So you go to Analyze Peptides, and there's a Libra section. As you can tell just even from looking at this screenshot, there's not that many parameters because they all live inside a condition file. And so Petunia has I think I showed you here, has a facility where you can actually generate your condition file and then specify it when you run Libra. You say, use this one file. And when you run, this will run, and then on the output, you'll see, again, this is the green one, the PepXML viewer. You will find that you are able to see for each peptide, for each PSM, you'll see the different Libra 114, 150, the masses, the channels that you used. You don't have to use all 4. You can use 2 or 3 or 4 or 5 or 6 if you use the 8 reagents. It will only display to you the ones that you chose to analyze. You can either view the raw values, the absolute intensities, or normalized. When you choose normalized, whichever channel you pick to be your reference will be 1, and then all the other values will be scaled to that, which is a much easier way to compare things than these big numbers. And at the protein level, there's no pretty way to display this, but you will see that when you look at the older XML viewer, you'll have a Libra result. It'll have a parenthesis with the number of peptides I used, and then the ratios for the different channels. And it also automatically produces a tab separated values file, so something you can import to Excel, where you have, for each protein, its ratios with errors, and each peptide for that protein, it's their ratios and errors, and whether yes or no they were used in the quantification for that protein. So you can get all this information out and then use it. Again, this will make far more sense once we actually start clicking and running the software. But